Oh, Michelle's here. Did the link work?
Yeah.
I didn't use the link.
Oh.
Okay, so guys, we will get started here. Welcome to our first live session. Obviously, thanks for dealing with technical difficulties. I'm not sure what happened there. My audio just stopped working, even though it tested like it was working. What I will ask you to do is to, yep, good call, is to just mute unless you have a question, okay? If you do have a question, just unmute yourself. You can ask, or you can use the raise your hand function. If you don't know where it is, I think just search around a little bit. You should be able to find it there. Just raise your hand and then it'll tell me that you have a question, and I can unmute you and you can ask your question when you are ready. Before we get started, does anyone have any questions they want to ask right away?
No.
No, we're good.
Mr. Nilan, do we need to join the folded that account thing? It was
no.
in your PPT.
So, yeah, I just re-downloaded it, and for some reason the application isn't working. So I'm gonna wait until, after this lesson, I'll play around with it a bit more and try to see if it even works. It's kind of an old program, so it's possible that it's not been updated or has stopped working. So next lesson, I'll let you know if that's something we need to deal with.
Shema?
It's on the first slide of the PPT.
Oh, sure. Yeah.
I'll show you.
Oh.
Okay, thanks.
Yep. Anyone else? Okay. So just again, like try to participate, guys. So if I hear your voice or if you have a question, please ask it so I know that you're alive and you didn't just join this and go back to bed or something. Make it useful. Okay. Yeah, let's begin. So first of all, we are now into week 3. So week 3, what I'll do once this is done is I'll put up the video here so you have it for future reference. We're dealing with 2.4 proteins. If you haven't caught up yet, you'll also know that I uploaded all of our previous videos to YouTube. So I'll start doing that, I guess. Might take a bit of time, but I think it's easier for everybody to get to. I haven't put up the HL ones yet, so I'll put the HL ones up tonight. And then please download this file, the topic questions and notes, which will look almost the same as all the other ones like this. Okay. And I've asked you to do like question 1, 2. These questions are really silly, so don't answer those. 3, 4, 5. And then I think it's 6 to question 10 after the end of today's lesson. Okay. So those are for after the lesson to do some practice. Everyone can see my screen, yeah? Or like, yes, yeah, screen is visible?
Yes.
Awesome, awesome, okay. In that case, let's try this. Okay. Right. So, proteins. The nice thing is, even if you haven't caught up yet with all the other stuff, this sort of stands alone anyway, even though it kind of builds on the metabolism, monomers to polymers kind of thing. But you can start and do this lesson and then go backwards and do carbs and lipids if you haven't done those already. But this is going to focus on a few things that we did last year with transcription, translation, those kinds of things. So how do we build proteins? And then what we add on in DP is some specific proteins and their actual functions, okay? So for all living things, there are so many different proteins that are necessary for life. We will look at the examples in lesson 2, or I guess the second lesson of this topic. But for today, what we're gonna focus on is how are proteins actually made, okay? So why not just jump into it? Okay, you can take a look at these later, but I'd like to just, yeah, thanks for that. What you'll find here is some, one of the old understandings. So this, when you see red here, you know that typically we have, our slides have the blue top thing here. But if you see something in red, it means it's something we have done already. So we've done this 2.1 in the SL topic. All right. You should have seen this slide previously of how a ribosome condenses two amino acids into a dipeptide, forming a peptide bond. This is really what this whole today's lesson is all about, is about putting amino acids together. Okay, so you do need to be able to, if you haven't already practiced, to draw this one peptide joining to another peptide through a peptide bond. This is a condensation reaction, okay, meaning that a water is formed. So you get this water being kicked out over here. And this forms a dipeptide, but we would also, if you're producing a polypeptide, this reaction would happen over and over and over and over, creating a very long chain of amino acids, okay? I'd like to see myself here like this. Ah, there we go, that's better. Oh, now I can see Jay, wonderful. Can I get rid of this? Save, okay. Okay. It just makes me feel like I'm actually speaking to someone if I can see your faces. Instead of talking.
Oh, we don't speak.
Yeah, well, if you could, that would be awesome. Okay, next understanding. There are 20 different amino acids. So of all the proteins that are built in the world, we're going to see that there are like infinite different combinations, but every single protein is built out of these 20 building blocks. There are only 20 of them, okay? But because you can order them in one, any number of combination, and two, because the size of the chain is variable, there's like unlimited possibilities for how a protein can be made, okay? So what is the most important kind of player in this regard? It is the ribosome, right? We know the ribosome to be the area of protein synthesis, the molecule that's responsible for this. So ribosomes are going to be where the one mRNA is going to come to, and then where tRNA is going to bring the amino acids, but it's basically the place where everything comes together, okay? So the term we should use is where polypeptides, meaning many, and peptide meaning an amino acid, so where multiple amino acids come together, synthesize meaning to build one molecule, right? Ribosomes are responsible for that. So the formation of this bond, where does this bond take place? It doesn't just happen magically out in, the middle of wherever, although maybe
covalent bonding.
Yes. Although once upon a time, right, when we say like the origins of life, like at some point, yes, it did happen magically a little bit, but now any protein that's made needs a ribosome to help make the bond happen. Okay, so it needs this sort of like positioning. So like all the stuff that's responsible has to come to this one place, that's the ribosome, and then we can put it all together. Now we will, this process of like replication, transcription, translation, we'll go more into detail in that next year when we do the genetics unit. But for now, what's important is the ribosome is the sort of the location where polypeptides can be formed. Okay, if we move ahead, here are, again, we've seen this before, I'm sure, the different R groups for the amino acids. So every single one of these, 1, 2, 3, 4, 5, these are different kind of variants of the amino acids. So again, you bring these into a.
What happened? Stop.
Stop. 1, 2.
Am I back?
Oh no. I thought I was stuck because no one was moving.
Yeah, I was.
But like,
hello?
like I can see Jay and Michael, like you two, the screen is like changing constantly. It's like for when Jay speaks, it's Jay. Then when Michael.
All right, we're good.
Coming laggy.
Yep.
Oh, no.
Yeah.
All right, so we were here. Oh no.
No!
We were here on.
Yeah, we can't hear you now.
Yes.
But we can see you. So.
What about now?
Oh,
We
hi.
can hear you.
I'm in confusion in the highest order right now.
So there's anyone
Jensen.
with his audio.
Can
Can you hear me now?
Yeah.
Okay, so it's when I share the screen. Let me try this, okay? I'm going to share the screen again. Can you hear me now?
Yeah.
Yes.
Okay,
Yeah.
You
All
need
I'm
5G.
going to try. Just let me know if it cuts out.
Okay.
I mean, if it doesn't work, you can get someone to share the screen and then.
True. Okay, so we were here, we're talking about the 20 amino acids. So in some combination of these comes together to make, William can't join, god damn it.
Wait, does Zoom have like a limit for how many people can join?
No.
Okay. A limit.
Okay, I'm gonna keep going while we wait for William.
Wait, but why is there 21 though? There's 21 amino acids in this picture.
Thank you.
Come on.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15,
Knock it off.
16, 17, 18, 19, 20.
So one part of this that we need to look at is that, yes, these 20 naturally occurring amino acids are needed to build all these proteins, right? Some of them, I mean, come on, William, figure it out. Some of them we can build by ourselves, like animals can create, like synthesize them on their own, but other times like for example, we have something called essential amino acids that we have to get from our food sources. We don't produce them on our own, so we actually have to consume them. Additionally, you'll notice here at the bottom that it says there are 22 amino acids. These are like newly sort of discovered, and they're actually not like from the basics. So if you wanted to just say there are 20 amino acids, that's fine, because that's what is normally accepted. But what's coded for by the genetic code, there are some that are like post-production. So after becoming a part of a protein, there might be some like modifications that take place that turn that into a different protein, so a different amino acid. So an example is this hydroxyproline. It's not a real amino acid from the genetic code, but after being modified, it becomes this different version. So it's really not, it's not like, if you want to call it like an OG amino acid, right? It's like it's something that comes later on with some changes that take place. So we can find
What modifies it?
What modifies it? That's a good question. I would assume there are some other proteins that come along after. So for example, the proline becomes a part of this. This is collagen, which you saw as, no, you didn't see, sorry. We're gonna see it later. So when it becomes part of the molecule, there's probably some other proteins that it'll cut little pieces off or add a few little details to the molecule to make it slightly different than its original kind of like raw version. Even if you think of like in the Golgi apparatus, you know how we said like post-translational modification, right? Like little changes happen to make it a bit better of a molecule than it is just the way that it comes out from translation. So these are very new discoveries, these new amino acids. But at the very basic, in terms of the genetic code, the pure genetic code, we only have the 20 that we see here. Okay, so what do you want to be able to notice here? We want to notice that the structure of these, right? It follows the basic structure from here, right? With the central carbon, amine group, carboxyl group, amine group. So if you look here, you have your amine group, you have your carboxyl group, and then you have whatever this thing is, right? This is your R. So R is like, you can see it's very different. It changes for all these 20. So you have 20 different versions of R, which means 20 different versions of amino acids. Now, as I mentioned, what's going on in the chat? Who's chatting?
What?
William said he sent the request. Oh, sorry. Let's get William in here.
Oh, that chat.
William, accept. Okay, and then let's get him in here.
Let's get this party rolling.
Okay. All right. Come on in, William. This is very strange. What? Get out of here. Why can't I? Oh, there we go. Okay. So the question here, why are there infinite possibilities of polypeptides? Because there are three things. Yeah, you should be in. What is this problem? I invited you. Yeah, these three things, right? So one is theoretically, there's no limit to how big a protein could be, meaning how long a protein could be.
60. Nice. I am not doing that.
There's no limit to like how many amino acids could be in a protein. That's already pre-coded for, but like there's no like a protein must be X length, right? The next thing is that there are 20 different amino acids. So the combinations can go on and on and on and on. And the order of those combinations can completely change, which gives us always new proteins because the interactions of the amino acids that are beside each other can always change. Okay, so because of this, that means there is really no like end to the possibilities. You take any of these, you mix and match them beside each other and create, think of it as like, imagine if you were making like a bracelet, and I would probably show you this if I was in class, right? You have like a thread and you have beads and you have 20 different colors of beads. And we say, make any length of bracelet you want in any combination of all of these 20 beads. Well, then the different types of bracelets you could make are infinite, right? Because there's no limit and there's no order to what I've asked you to do. You could take one color and make it a million long bracelet of just that single color. That could be a protein. You could take a million length of one color and add one different color at the end, and that's a completely different protein suddenly, right? So that's this infinite possibilities idea is really important for proteins, okay? If you did the math on this, for example, if a polypeptide contains 7 amino acids, right? So that means it's 20 to the 7 possibilities. Then you have a potential of having over 1.25 billion. Can you guys see this?
Yeah.
Is
Yeah.
William calling me?
Mm-hmm.
Who calling you?
What?
William is calling me.
Oh
Oh,
no.
no, we can't.
I don't know how to get him in. I already invited him. Okay. Let me check one more time on William. Invite. He's been invited. Wait, Yushan, you're in, right? I invited you.
No, but like even if you invited, you still had to get the code.
Okay. What did you do?
I got the code.
Why can't he just get the code then?
Just text a Chinese number. Like if, because you should text my number, but like I didn't see it, but I probably figured she'd text somebody else.
Oh, true.
Like he can do that.
True.
So
What the Bahamas?
wait, I'll text him my number and so he can text it
Yes.
and get the code.
Okay, am I still good? Can you still hear me?
Yeah.
Oh, thank God, because it just told me I was unstable again. Okay, so infinite possibilities for polypeptides because of these three things. Can be any length, up to 20 amino acids in any combination and in any order.
So basically it's like 20 elements, but you can just put how many ever you want.
How many ever and in whatever order, right? It doesn't matter the order. It doesn't matter how many.
Okay.
Okay. Now.
Okay.
Whatever. Okay. I don't want that to be like that. I want this to.
Mr. Ilan, in your PowerPoint, it says up to 30,000 amino acid, but is it like really no limit, or is it?
Yeah. So that is the largest protein that we know of.
Oh,
is that
so it's possible in the future that protein suddenly form like, more than 30,000 amino acids.
Yeah, like it hasn't been discovered yet, but I guess it.
And then found by like some scientists whatsoever.
Yeah, exactly.
Okay.
So as far, stop that. Okay, as far as we know, yes, we only have, that's the biggest one that we know so far.
Okay.
Oh, yeah, thank you, good question. Okay. So yes, we mentioned this, ribosomes have the site of polypeptide synthesis, but they need a template. So basically saying like, how do you build the protein? So meaning, how do you know the order, right? How do you know what order to put this protein in? Because if I just give you the beads and say, okay, make this bracelet, then you can make it however you want. You can decide the order, you can decide everything. But if you want to build let's say a bracelet that has a specific function, like if you want to make a protein that actually works and does something you want it to do, then the order is really critical. You need the order to be correct, right? So where does that order come from? The order comes from something called mRNA, right? And we saw this last year, mRNA, or messenger RNA, right? So messenger RNA has the information you need to build that protein, and that's shown here, this kind of greenish tealish color, okay? It's a code of, I actually thought that was Michael sitting there listening. That's pretty good. Like, that's good. So on this code is the information, and that information comes in the form of codons. These are all terms that we heard last year, right? Codons are triplets of bases. I don't know if this is becoming familiar or not, right? So triplets of bases in like 3-letter forms, and each 3-letter codon helps to code for one amino acid to show up, right? Mm-hmm. So codons on the mRNA tell us which corresponding amino acid should show up.
ACTU thing, right?
Yeah, exactly. So once upon a time, we had, I think I have it. Oh, no. There's like a whole, like a grid, like a diagram that has like 3 letters, and you can like trace across to see which amino acid corresponds with which triplet. Well, again, we're going to save that till later. But the main thing that we want from this is this idea that we know as, it's called the central dogma of biology. And I'm sure you've heard this before, or you've heard me say this before, okay? The central dogma of biology says this. It says that DNA becomes RNA, which becomes protein. Okay? DNA to RNA to protein. What are we saying? We're saying that all of the information, okay, no, let's go backwards. Let's start with protein. So protein, everything we need, everything that makes up living stuff is built out of proteins, right? Like you need proteins as the like material for building life. But you can't just have the materials. You need a way to put the materials together. So how do we know how to put it together? We need instructions. So the instructions come from RNA. RNA is the instruction for individual proteins. But where does the information for these individual proteins come from? It comes from a huge library. You could say like the textbook or the manual, okay? This is where you have all the instructions, okay? It's kind of like a manual of how to build every single protein ever in a given organism, let's say. Okay, so how do we, why is this important? Why is this, or sorry, what is the relevance of this? The relevance of this is that information for building any type of protein is in your nuclei in the form of DNA. But you can't build all proteins all the time. You can't build all proteins at once. Any given cell only builds a certain type of protein based on its specialization, right? Like, for example, let's say epithelial cells on your skin surface, they need to grow, like they need to build proteins like hair so that they come out and so you can stay warm and blah, 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 right? That information for building the hair is in your skin cells, but it's also in your white blood cells. It's also in your nerve cells. That same information exists in those cells, but you don't want your nerve cell to start growing hair.
Why not? It sounds cool.
I guess it could be cool, but it's a little bit weird. So you have to control this, right? And HLs, we talked a bit about gene expression when we did the plant stuff. So you want certain things to be turned on, certain things to be turned off, so that you don't build all of the proteins. You only build the proteins that are needed for that cell to do its job. So yes, every single cell contains all the DNA, but you only transcribe, right? Transcription, we only kind of write out some of the instructions when they're needed. And then those instructions that we transcribe, we will then convert into proteins. So here's my RNA over here, this strand there, and then that would have come from the nucleus. It's not seen in this picture, but it would have come from the nucleus as a small message, but the nucleus has all of the messages in it. Okay, so this concept, this central dogma of biology, this is something that will come up quite a bit when we get into genetics again later on. Okay, where does the messenger RNA come from? It comes from the nucleus. Here you are. Oh, this is what Michael was talking about, the blue boxes. So if I go, I know, I'll just delete this. Okay.
No,
So
I actually found a way to use the things. I can just use the school's account and use the online version. I just need to upload it to online, then it'll work.
Nice. Okay. So I'll stop making PDFs then.
What?
I did upload some in PDF format, but I guess.
Oh, yeah. You can just upload normal ones.
Okay. So here you go. Yeah, these are the processes. DNA in the nucleus gets transcribed into smaller messages as RNA, and then that leaves the cytoplasm. Leaves the nucleus into the cytoplasm, and that's the point where ribosomes will attach and start to translate that message into the protein or the polypeptide.
Mr. Neon.
Yeah.
RNA are proteins, right?
No, RNA is a nucleic acid.
Okay. So it's not made.
William?
It's
Sorry.
not made in the ribosomes or whatever. They just appear.
They are formed in the nucleus through a process called transcription.
Okay.
Yeah.
Wait, RNA, right?
RNA,
What's
yes.
the components of RNA?
So it's exactly the same as DNA, except remember what's the difference?
Oh, it's a
Ribose.
Ribose, right? Ribose sugar instead of deoxyribose.
Oh
And
my
then
God, how
another
do I
one
know?
is that T's are replaced by U's.
What's the only difference between mRNA and just RNA is that mRNA is just the copy made, right?
Actually, there is no, so RNA is just like a category. And then mRNA is a type of RNA. There's no such thing as just like there's nothing called just RNA. So there's mRNA, there's tRNA, there's rRNA. So the one we're talking about is mRNA. That's the copy, the little message. The M stands for messenger, actually.
Oh, so it's the same thing.
Yeah. So if you had RNA as the title, then there would be like 3 different types, actually more than that, but 3 different types that we know of, or that we'll learn. So messenger is a type of RNA. If we go here, actually here, you can see there's something called tRNA. So that's another type of RNA.
Oh.
We need to know what that is.
Later, not right now. Transfer. See here, it says it. Transfer RNA. Yeah. Okay, can you get rid of these numbers?
Oh.
What?
Chain. Thank you. Okay. So as you mentioned, yeah, there's 20 different amino acids. They can be combined in any order. And then we mentioned this briefly when we did the monomers section. So there are different characteristics to all those R groups, right? They can be polar, they can be non-polar, they can be positive or negative. Some of them have sulfur. So because of how every different R group is different, when you put them beside each other, they will react differently, right? Think of if, let's say we took the entire class, right? And I just like mix you guys up and I said, everybody hold hands and mash together. Again, if we were in class, this is what I would do. Yay, hold hands. Okay. Mash together, right? If I did that, there would be some people that were like beside someone they're comfortable with. There'd be other people who are like beside someone they really aren't super comfortable with. Or mash together. You'd be like face to face with someone you really don't like and you'd be like, oh, right? Awkward. Or you'd be face to face with someone you're cool with and you'd be like, yes, what's up? The R groups of amino acids work exactly the same way, right? So if you have this long strand, okay, and along the way there's different R groups, there's parts that are going to be attracted to each other, there's parts that are going to be not attracted, then like this end is attracted over here, and all this does is it ends up like folding the protein into a certain shape. And the shape is going to be important in its functionality, okay? So along the whole strand, because it's just amino acid, all beside each other, their interactions will determine how this string will fold up. Okay, so that's what this red circle is circling. The properties of the amino acids determine how a polypeptide folds into a protein, right? And what is that? What properties are we talking about? We're talking about the differences in each amino acid's R group. Right? Think of it. If you have like 2 amino acids attached together, one is positive, one has a positive R group, one has a positive R group, then they're not going to like each other, right? Opposites or same charges don't like each other, they're going to repel. And that's going to cause a bend in that point in the polypeptide chain. Versus if one has a positive R group beside a negative R group, then they're going to attract to each other and suddenly that chain comes closer together. Okay?
Okay.
All right. And this is actually our last slide for the
that?
day.
That's
Last slide
already
for today. Okay. Really important though, this box says that this is only HL required material. And it's not actually in this topic, it's in topic 7. So SLs, if you don't know it, it's not the end of the world. This is more of like a preview for HL students. But all this is showing us is that proteins have 4 levels of organization. Okay, so we call them primary. What's going on here? Primary, secondary, tertiary, quaternary. Right, just meaning first, second, third, fourth. Okay, so we can show it with a 1 with a little. Oh, I can't find that symbol. Okay. Anyway, you get
the little
primary.
what?
It's like the degree sign.
Oh.
Okay. So all we're saying, this is good. It's a good thing to know in terms of the organizational structure of a protein. The primary structure is really just the polypeptide chain. So what determines the primary structure is the order of amino acids, right? The order of amino acids. So if I asked you how do you know what is the primary structure of a polypeptide? Well, you would say, oh, I need to know like what's the order that amino acids go beside each other. So it's determined by, it's determined by the mRNA, which tells you what the order of amino acids should be. So the polypeptide chain is considered the primary structure. The secondary structure is determined by the initial folding. Okay, so the initial folding, not even folding, but interactions between amino acids that are close by. So there are two types of interaction. We call them alpha helices, which is plural for helix, and beta pleated sheets. Now, this sounds absurd, I know. Just give me a second. Yeah, I know. Okay. All right, let's zoom in on this picture here. All right, here. So, that is not very explanatory. Let's draw it. Okay, so an alpha helix is just a spiral. Okay, alpha helix.
Wait, your audio just cut.
No.
Like just say the alpha part again, because now I can hear you.
Okay, alpha helix is just basically a coil. Okay, let's do this. Let's get rid of that. Okay, alpha helix. Right, is just a coil, right? So it looks like this one over here. So meaning that some of the interactions, like a few amino acids are sort of attracted to each other, but it creates this sort of spiral shape, versus a beta sheet, beta pleated sheet. Pleated means to be folded. So beta pleated sheet looks more like a zigzag. Oh no, that's ugly. Beta pleated sheet is done. Oh no, oh thanks. That. Okay, so some of these very like nearby interactions will either produce a spiral or a sort of folded shape. Okay, it kind of gives you like an equal attraction or repulsion between close by amino acids. So these are both these are both secondary types of structure. Now, some proteins will actually stop at that point. Okay, they will stop and just be either a couple of helices, a couple of sheets, and then that's done. And these are usually fibrous proteins. They're like elongated. So things like, we'll see this next time, like collagen or spider silk, some proteins that are really long and stretched out. But most proteins that have a functional element, some like that actually do work, that are going to be globular proteins. Okay, so tertiary proteins are the three-dimensional shape. 3D shape, meaning after forming some beta sheets, after forming some alpha helices, then so imagine if this one, okay, we had like a spiral over here. This is really not what I want to be using right now. And you had a zigzag thing over here, right? And then like the zigzag attracts to the spiral and it becomes more like a blob, right? So the blob part is my tertiary structure. So when like the alphas and the betas sort of interact with each other, that's your tertiary structure. Again, if I was at school, I would have a pipe cleaner or something, and I'd be able to actually show you this properly. Okay, but the 3D shape, we could say that it's globular, right? Globular, like a glob. And the last is quaternary. For now, let's just say that quaternary is like when many tertiary, wow, proteins interact. Okay, so what you can see here is like, you see this pink, green, yellow, blue. So there's like 4 of these interacting with each other. All right, so when they come together, they can form like it's like a Power Ranger. What is that thing called?
Megatron. What?
Megazord? What is when like the individual like
What?
things, they come and form like a big one?
Yeah,
Yeah.
too
That-
old.
Thing from Wreck-It Ralph, the villain.
Maybe, I don't
No.
know. I can't think of the name. Like they have their own machine, but then they can like come together to form like a
Power Rangers assemble!
Yeah.
Red Ranger, Green Ranger, Big Ranger, Yellow Ranger, Green
So-
Ranger.
Yes, exactly.
Okay.
So
Is
this blue one like might be able to do some job by itself, but when it comes together with these 4, it can do like another different job. So yeah, we can say interaction between multiple polypeptides or the addition of some other little like other molecules. So the example they use is hemoglobin. So hemoglobin has 4 tertiary proteins working together, and then it also has something called heme, which is this like iron-based. Is he in here?
for Jack.
No, like
Yes.
yeah,
Let me see.
the heme group is there. Okay, that was very stressful, but I'm glad it worked out somehow. That's it for today. Okay, we will pick up here tomorrow. How's everybody doing? Is there anything I can answer right now for you before we end off?
Can't think of anything.
Anyone?
Yes.
For any like bond between atoms, do you just call it covalent bond?
Oh my god.
As far from what we've learned, yes, most of the bonds we've learned are covalent, but you should know like the specific names. For example, for proteins, we say that it's a peptide bond. Sugars are what?
Glycogenous bond.
Glycosidic.
Glycogenic bond. Or something.
Glycosidic, good. And then, tricky one. What's the one between glycerol and a fatty acid? It's the name of someone that used to go to our school.
Ester?
Yeah,
ester bond.
ester bond.
Yeah.
Ester
How
bond.
do I know this?
Yo, what? I never got the story of what happened to Esther. Anyway, not important right now.
XD.
Did I learn something from e-learning?
Boom.
Mr. Nilon, can you re-explain the alpha helix thing kind of?
Yeah, it's really just, very simply, like the very first like interactions between close amino acids is either alpha helix, meaning it'll make the like straight chain turn into more of like a coil, like a spiral, or the interactions will make it go in like a zigzag shape, like a folded piece of paper. So those are just 2 types of interactions between like really nearby amino acids.
In primary structure.
That's a secondary structure.
Oh, secondary. Okay.
So the primary is just the straight chain. What's the order of the amino acids? Then after that, you get a little bit of interactions like folding like this, like folding along like a single plane, right? Or spiraling. And then you get blob, it turns into a blob. That's your tertiary structure.
Oh.
These are proteins, right?
Say again.
These are proteins, right?
Yeah.
It's all about proteins.
Mr.
The
Nuan.
topic is even like just like
Yes, Jansen?
Just to confirm again, this won't be on our SL, right?
Correct. This won't be SL material.
Oh,
Shema?
wait, really?
Oh wait, really?
Just this slide. Just this slide only.
Ayo.
Oh,
Mr. Nilang,
I'm really sorry to interrupt you, but like the math, like HR class have a video call right now.
Yeah, for sure. Also, I was gonna say, Mr. Yubin messaged me. I think we'll start tomorrow at 3:20, because it sounds like your econ goes till 3:20 usually. Yeah. Okay, so let's do that. I'll send
Maybe you can just delay everything to 3:30. Like sounds small.
Sure.
Like,
If it works for people, yeah. Anyway, we can only do 40 minutes, so we'll be done 3:40 to like 4:20. 3:30 to 3:30 to 4:10, I guess.
Okay.
Okay, thanks for your patience, guys. I don't know what changed, but the last bit of this worked pretty well. I will upload this, or I'll merge the two pieces together and upload it onto YouTube. Yay! And I'll put it on
Click here to
screen as well.
subscribe and click here to comment.
But also, like the pixels are really like sometimes you can't see things. Like
Yeah, so I'm gonna keep the resolution. I did something dumb because I was trying to rush for Jay, who didn't do any videos. So I just
Before the
made
video
it really
ends.
garbage quality for the first ones, but this one I'm gonna try to make it better quality.
okay.
Thank you guys for watching this episode of Biology is Fun. Subscribe and comment and like below. You know what's the drill. And as always, stay awesome.
Okay, see you guys.
Okay, thank you.
Keep it
Thank you,
real.
Mr. Nilang.
Thank you, Mr.
Thank you,
Nilang.
you.
Mr. Neon.
Thank you.
Goodbye, Mr. Nilan.
Bye, guys.
Bye.